So now let's move into Kingdom Animalia. So this is the kingdom that we are a part of, and by far it's probably one of the weirdest ones yet. So here's the duck-billed platypus. It's an organism that has a bill like a duck, fur, and it is a mammal. It feeds its young with milk, yet it lays eggs. It has a poisonous spur on its back feet, and it detects its prey by detecting their electromagnetic field. So it doesn't even need to open its eyes to hunt. That's weird. All right. So that's where we're headed as we head into the animal kingdom. So what characterizes an animal? I want to take this first recording on the animal kingdom and just talk about the background information, introduce the 9 representative phyla of the 35 phyla in the animal kingdom, and then we'll subsequently go through each of those 9 phyla in a little bit more detail so we understand the diversity that we see in the kingdom that we belong to. So if you're an animal, you're eukaryotic, you're multicellular, you're heterotrophic, all right? You lack cell walls, and that's going to distinguish us from the fungi right there. We already are familiar with the gametic life cycle. And then finally, the zygote goes through a common development in the animal kingdom, and that's through a blastula stage and a gastrula stage. And what that means is if we look at the development of that zygote, remember that zygote is that first cell that results from the fusion of sperm and egg, and it is now diploid. So it starts to divide mitotically and goes through a 2-cell, 4-cell, 8-cell stage, and ultimately forms this hollow ball of cells. And that is called the blastula. That's very, very important because at this point is when the genetics of the animal are setting up the body plan. And so, but every animal goes through this very same development at this early stage in the game. Now, the blastula will continue to develop, kind of indent, fold in on itself, and form what's called a gastrula. And so that gastrula then establishes some of the basic tissues from which the rest of the animal will develop. So this is important because all animals go through it, so it's a commonality in the animal kingdom. Now, the 9 classic phyla that we study in the animal kingdom are listed here. Realize these are by no means all of them. There are 35 phyla in the animal kingdom. These are the ones that most textbooks cover, and we cover these because, one, many of these are stuff that you're familiar with, that you interact with, or may be important because they cause disease or are important for a food source. But also they'll show a couple developmental trends as we go from the simplest of animals to more complex animals, and finally studying kind of chordata, which is the phylum that we belong to. Now, once again, remember phylum, let's go back, dumb King Philip, phylum is a very high level of taxonomy. So there are many, many species within each of these phyla, and there's great diversity among those species. So again, we're just skimming the surface, just so you have some feel of kind of how animals can vary and how we attempt to organize them. So we're going to look at porifera, the sponges, cnidaria, which are the hydras and sea jellies, sea anemones and corals, platyhelminthes, which are the flatworms. They're both free-living and parasitic. We'll look at nematoda, the roundworms, mollusca, snails, slugs, clams, octopi, and squid. Annelida, which are the segmented worms, the earthworms. Echinodermata, which are the sea stars, sea urchins with they have what's called radial symmetry. Arthropoda, which are the crayfish, lobsters, crabs, spiders, ticks, and insects. This is the most diverse of all the phyla. It is the largest. There are more different species of arthropods than there are of all the other animals in the animal kingdom. There's over 1 million. So that's a whole lot. And we'll talk a little bit about why that is. And then finally, we'll look at chordata, which is the group that we're in. So this is the fishes, the amphibians, reptiles, birds, mammals. Okay? So on this chart, you may look and see that there's a big split after we form a celum. And we'll talk about this idea of a body cavity as we go. And we'll also talk about this idea of symmetry within the body. We see that porifera, the sponges, have no true tissues and no true symmetry. And then with cnidaria, we see radial symmetry, and then we move into bilateral symmetry. Now, as we move up to celum, this idea of having a body cavity, we actually look at developmental differences. And that's the difference between the protostomes and the deuterostomes. and that is when you form a blastula, and then goes through gastrulation, how that gastrula forms and what the pore, the blastopore, that opening on the bottom becomes, whether the mouth or the anus, is huge kind of from a developmental point of view. And so that is the division that we see here from the protostomes versus the deuterostomes. And that's what throws echinodermata in on a more related branch with us than the other organisms. Whereas echinodermata, the sea stars and urchins, they don't look much like the chordates at all. But in fact, because that developmental characteristic is there and shared, because it's so fundamental, it pushes them more towards us from an evolutionary point of view. And so you can see that in the sea stars, that gastrulation event, then going on, the sea star develops into a larvae that then eventually becomes an adult sea star. Now, the other kind of groundwork that I want to lay down in this particular recording is when we talk about body types. And so we're going to look at symmetry in body types, asymmetric in the sponges, radial symmetry in some, and then mostly bilateral symmetry. So if you're radially symmetric, basically any given diameter through the organism, through its central axis, will give you two halves that are the same. So some sponges, the sea jellies, the hydras, these are radially symmetric. Bilateral symmetry means that you cut through a central plane to get two mirrored halves. So bilateral symmetry is kind of the most common form that we'll see in our representative phyla. And it means that you've got basically a head. And when you have a head, it means that you have cephalization. So bilateral symmetry means that you've got one end that has a clustering of sensory organs and nervous tissue, right? i.e. a brain. So that the fancy word for that is cephalization. And we're going to see that as we move the flatworms, the vertebrates, the nematodes, the arthropods, chordata are all going to see cephalization. So we're moving in that direction. Now that also allows us to set up a vocabulary to orient kind of the anatomy of an organism. And this is more stuff that you'll learn if you go on to take anatomy and physiology. I just kind of want to lay out some of the basics here as we talk about it, and I want you to be familiar with it. And those are now, if you have experienced cephalization, you have a head. It means the end with the head is called the anterior end, and then the end opposite the head is called the posterior end. So anterior versus posterior. You also then have a dorsal versus a ventral side. The dorsal would be the back side in relation to the head, and then the front side or bottom side would be the ventral side. So you have a dorsal-ventral opposition there. And then finally, the sides along the anterior-posterior axis are called the lateral sides, left and right. Okay, so not a whole lot of new vocab there, but important to know: anterior, posterior, dorsal, ventral. Now, a lot of people want to immediately apply that to humans, and with humans you can, but you have to think about it a little differently because we are actually bipedal, and one of the very, very few organisms that display bipedality. And so with that now, what we've done is taken our ventral side and actually moved it, gone vertical with it. So our front side is really ventral, and our back side, or on our back, is dorsal. There's more. We talk about distal and proximal, superior, inferior, medial, and lateral as you get more into human anatomy. And we're not going to cover that in this class, but you are headed for that if you're headed for A&P.